I want to welcome you all to Wednesdays at the Center at the John Hope Franklin Center. Today we have Reshma Nargund. She is a PhD candidate at the Nicholas School of the Environment and is a current Global Justice and Equity Fellow here at the John Hope Franklin Center. She is passionate about studying the effects of air pollution on human health. She is interested in exploring environmental justice issues associated with the disproportionate burden of environmental pollutants in developing countries and low socioeconomic communities. She is co-advised by Doctors Susan Murphy and William Pan from Duke University. Please join me in welcoming Reshma.
Sharon, and thank you all for coming. I'm excited to talk about my work. But before that, by a show of hands, how many of you have heard of this term epigenetics? All of you. That is great. So I don't have, sorry? You just heard the term. Okay, so let's dive right into it. So what is epigenetics? We all seem to understand the DNA. We all seem to understand genetics. Very simplistically put, there are these genes that go on to define what kind of eyes we have, how tall we're going to be, or how short we're going to be. But there's this whole other layer to genetics, which is called epigenetics. So the thing to remember is that every cell in your body has the entire DNA to make a human being. Or in the case of a cat or a dog, the entire DNA to make a cat or a dog. But why is it that a cell in our skin behaves so differently than a cell in our heart? And that is because even though the entire code to make, the entire genetic code is present in every cell, only, for example, in skin cells, only those genes that are associated with skin function are turned on, and everything else is turned off or switched off. Similarly, in a heart cell, only those genes that are responsible for heart function are turned on, and everything else is turned off. So that is epigenetics. Now, let me give you some very fun examples, some fun and some not so fun examples of how epigenetics plays out. I find this example so fascinating. Now, we know that the worker bee and the queen bee, again, they have the same DNA. They have the same genetic code. But what happens is, during the first 3 or 4 days of the life of the larva, they're given the same kind of food. But after 4 days onwards, a few bees that are going to be queen bees are given a different kind of diet than the rest of the worker bees. So they're fed this thing that is called royal jelly. And because they have a different diet, they go on to become entirely different creatures. Because look at the difference. This is the queen bee, this is the worker bee. Both of them are females. The size. The queen bee is so much bigger than the worker, one. And the queen bee is able to have more eggs. She has an ovary that is functional, whereas the worker bees are not able to hatch eggs. Importantly, look at the lifespan. This is 3 to 6 weeks, and this is 2 years. Imagine that if something like that were possible in human beings. Some of us would live for a few years, and others would live for hundreds of years. So this is fascinating, all of it happening because they are fed different diet, and then different genes are switched on and different genes are switched off, right? Okay, now this is an extreme example. It's quite fascinating. But in human beings, there is this unfortunate natural event that took place, well, not natural. During World War II, there was blockade of food delivery to parts of the Netherlands, and they basically suffered a famine for 3 to 4 years. Because in the Netherlands, there had already been very good record keeping. The hospitals were already maintaining really good records at the time. There's this cohort called the Dutch famine cohort, and the people that were exposed to this famine have been followed for generations together. So children of women that were pregnant during the famine, they're of course adults now, um, older adults in fact, and it has already been established that they have a higher incidence of diabetes, obesity, and cardiovascular disease. So what does this go to say? That during pregnancy, exposures during pregnancy, environmental factors during pregnancy can go on to have these effects that can last a lifetime. Not only that, the next generation, that is grandchildren of women that were pregnant during the famine, there has been some data to show that they are born smaller at birth. They have low birth weight because their grandmothers were pregnant, the famine. So the grandmother's experience is going on to have an effect on the grandchildren. So this was the Dutch famine. So we saw some examples of how it works in the animal kingdom and how we have some data about how this works in human beings. Okay, now coming back to pregnancy. Look at this graphic here. We're starting from one cell out there. At the beginning, there's the embryo. There's just one cell. It goes on to divide, and we basically have a human being from one cell. Towards the end, we have a little human being. Cells are dividing, of course, but they're also differentiating. What that means is they're going on to become heart cells. They're going on to become eyes. They're going on to become skin. And all along this time, genes are switching on and switching off. And there's a synchronicity to it. There is some genes need to be switched on exactly at the right time and others switched off exactly at the right time. And there is a play tag during pregnancy. This event happens, then the next event cascades, and there's a set of cascading events. So now it stands to reason that exposures during pregnancy can go on to have an effect on this, basically this mechanism of switching on and switching off of genes. So this is where I come in. This is what my study looks at. Then I come from India. I have little nieces growing up in India, and if you ever look at the AQI scores in India, if you look at the US, it's green and yellow. Sometimes it is red because of the wildfires coming from Canada. If you look at India, it's always red and purple. And that's the case a lot of places in China, a lot of places in Southeast Asia, very high levels of air pollution. Of course, here's some data. There's $8.1 trillion in health spent globally because of air pollution. Interestingly, or importantly, low-income countries are more affected by the effects of air pollution because they have more air pollution and they have less mitigating resources. Now that was globally between countries, there's a case of exposures to air pollution, but also, also within a country, there can be disparities. For example, in the US, the EPA has found that low-income communities have higher exposures to air pollution, not just to air pollution, but to all sorts of chemicals, because maybe they're living closer to the roads, they're living closer to some industries, and there are less trees in their neighborhood, less ventilation. So the point I'm trying to make with this is the place of birth is important, exposures, and it is important to lifetime outcomes because just because of where you were born, you could have these outcomes later in life from where you were born. So that's what makes this an environmental justice issue. I wanted to show this picture. This picture is, I used to live 2 kilometers from this place in Bangalore, and this is a lake. The lake caught fire, and it has been catching fire for the past 10, 15 years. It keeps catching fire, And it is a cocktail of sewage, chemical mixtures, and what have you. And you can see that the plume is engulfing these high rises here, where I'm sure people live, young children live, and we are living amidst all of this. And we don't even care that much because this is happening to us every day, like you cannot worry about it too much, and people have sort of accepted it as a byproduct of high economic growth. Again, talking about disparities between different parts of the world here. Right. What has already been established? We already know that mother's exposure to air pollution during pregnancy can result in these birth outcomes. Low birth weight, preterm birth, stillbirth, poor lung function, and childhood asthma. We also know that low birth weight is associated with a host of non-communicable diseases later in life, like diabetes and cardiovascular diseases. So although we know these outcomes, we don't know the exact mechanistic pathways that lead to these effects. So one of my hypotheses is that maybe the way genes are expressed, the way genes are turned on and turned off during very early life, during pregnancy, is leading to some of these effects. So I have to talk a little bit about, okay, I've been talking about genes switching on and switching off, but how are they switched off and switched off? So if you can think about the structure of the DNA, it's really, think about it as a spool. That is, it's a really long string of code that's tightly wound up in, let's say, like a spool of wool. And the more tightly it is wound up, the more switched off that part of the DNA is. The more open it is, the more switched on, because it's simple. If it is open, all the other set of reactions that need to take place can take place. That is one of the ways of epigenetic mechanism. Either it is very tightly bound or it is loose. If it is loose, all these enzymes and chemicals and what have you can access it, and then the gene can be expressed. So that is one mechanism. I am not studying that mechanism. What I'm studying is called DNA methylation. So this is the DNA, as this is a very famous picture. We all are aware of this picture. It's like a twisted ladder. The DNA is like a twisted ladder. And there are these, what we call these rungs of the ladder are base pairs. So methylation means adding a CH4 group. So basically, it's carbon and hydrogen. So for example, this here is a methyl group, a CH4 group. But if a part of the DNA is methylated, then it is switched off. Because, again, it's the same thing. These enzymes and precursors cannot access the DNA, and so it cannot go on to do what it's meant to do. So higher levels of DNA methylation means more switched off the genes. Lower the DNA methylation, more switched on the genes. So very simply, just keep that at the back of your mind, and I'll get back to it later. So what we did was we recruited around 215 pregnant women in and around the Triangle area that presented to Wakeman Hospital. And during their pregnancy, we collected socioeconomic data, nutrition data. We collected a lot of data during the pregnancy. Importantly, we also collected psychological and financial stress data. And so we have prenatal data, a lot of information about the mothers during pregnancy. And then at the time of birth, we collected cord blood samples. So blood from the umbilical cord. Now, the umbilical cord, the baby's genetic material. It is expressed from the baby's DNA. So The umbilical cord is supposed to represent the, simply put, those are the baby cells, you know. So we collected cord blood samples, and from those samples, we have the DNA methylation data that I just talked about before. So we have all this prenatal data from the mother, and then we have at the time of birth, we have DNA methylation data. But I am studying air pollution. Now, where did I get that air pollution data? We had not collected air pollution data for these women because we did not have like a personal monitor, nothing on them. So what I had to do was pull it from publicly available sources. Now, before I get into what kind of source I use, just a little bit about air pollution. These are the 6 main, they're called criteria air pollutants that the EPA sort of tracks. There's ozone, there's particulate matter. Particulate matter is basically dust particles of certain size. If it is 2.5, it is less than 2.5 microns. If it is PM10, it is less than 10 microns. There's carbon monoxide, lead, sulfur dioxide, and nitrogen dioxide. Sulfur dioxide is not that much of a problem anymore because during the 90s and the early 2000s, there were some policies implemented. It's kind of like a solved problem. The industries basically took care of it. Lead is also not that much of a problem in air. There is still legacy lead in soil, and sometimes the soil gets aerosolized, and that leads to lead in air. Nitrogen dioxide comes from vehicles, factories, and industries, but nitrogen dioxide and particulate matter are closely sort of related. As one goes up, the other goes up. And then there's ground level ozone. So I decided to focus on particulate matter because a lot of work has been done on the effects of particulate matter exposure on birth outcomes. So I wanted to see, okay, what's going on behind the scenes here? Can we see why these outcomes are coming up? So that's what I'm going to focus on. And that brought me to my first problem in solving this. In trying to answer this question, I don't have air pollution data. I don't have PM2.5 values that I got from the participants themselves. So how do I do it? So what I did was I geocoded. I knew their address during pregnancy, and so I geocoded their addresses. And can I assign a value to each of those geocoded addresses? So I looked at monitor data. Now, these are all women that presented to WakeMed Hospital. They all came from around WakeMed Hospital. They're kind of living close to each other. So when I looked at what monitors I can pull the data from, they were all being covered by 4 EPA monitors, which is okay. I can use it, but then they might end up having the same value, and I might not see a variation in their exposure value. But monitor data is really good data because these monitors are approved by the EPA, and that data I use it. So I decided to look at satellite data. Satellite data, now these satellites are circling the Earth every day, and they take pictures of the Earth every day. And the one way of getting PM2.5 value is like they take a, let's say they take a picture, and they take a picture basically of the air column, right? And based on the amount of particulate matter in that air column, there is light dispersion. So they calculate that light dispersion and somehow arrive at a value for basically every 1 kilometer grid on Earth. So there's a bunch of these sources, but I found one, Dr. Martin Randall's lab in Washington University. They have this data going from 2015 to 2021. Every 1 kilometer grid on the Earth has a value for all these years. So that's what I'm using. I also looked at chemical transportation models. These are models that make use of atmospheric chemistry. They ground truth it to monitor data. But unfortunately, I did not have this data available for my study duration. So we have data, we have women that were recruited from 2017 to 2020. And this data went only till 2018. So that data is yet to come out. the EPA has some data that they produce, but I don't have it for my study period. So I decided to use the satellite data. There's this, I just wanted to mention this. There's something called purple air. Have you heard of purple air, anybody? Basically, there are these very inexpensive monitors, they're like $200 or $250 that you can buy and install outside your home. And I looked at purple air data. Do you think I can click on this? Oh yeah. Okay, let me load it and then I'll show you the map. Right. So these are basically monitors that people bought and put outside their homes. And you can see that the air quality here is pretty good. Green and yellow means good. But let me zoom out and see if we can see some variation. Maybe LA region has a little more than here. I'll let that load. So I could use this data. So there's a little bit of variation here and there. The higher the number, the worse it is. Yeah. So yeah, California usually has some red spots because of certain weather patterns and certain geographical conditions. But I really want to look at China and India and see how different it looks. I'd have to zoom out quite a bit. So one of the problems, of course, with purple air is that it's not regularly, people have to buy it, so it's not uniformly distributed across, let's say, Durham or Raleigh. Also, there's a lot of individual variability between those sensors because it might be that somebody just put it very close to a grill, and on a day that they grilled, the levels were really high. There are some statistical methods you can use to take care of this, but I just decided to stick with the satellite data because it's uniform across. Yeah, so that's kind of what the world looks like. So this here is India. So if I were to look at India, one of the main first problems would be that there are such few sensors. On the other hand, if you look at the United States, there's way more sensors because people buy more of those. But it still gives you some sense. And I have seen some studies where they use this purple air data. They have to correct that data a little bit, but I've seen studies that use it. So these were all things that I found out in the initial phase of study. Any questions before I move out of here? Yes. So the first thing to remember is the way we define genes is if they go on to make a protein, that's when we call it a gene. That's the most traditional understanding of a gene. If this gene is responsible for the production of this protein, so that is one gene. And the length It could be, so we count it in terms of base pairs. So how many base pairs does a gene have? It could vary. It could be 100 base pairs. It could be 1,000 base pairs. It does not matter. It just depends on how much information you need to make a protein. There are, yeah, these are all sequencing technologies. Yeah. Well, so the no, so you have the DNA. Now the DNA goes on to make the RNA and RNA to protein. So there's sequencing technologies for the DNA, and then there's sequencing technologies for the RNA. And then there's a whole field of proteomics where people just study the proteins. That's a great question. It is not on or off. It's like a volume button. Either it is low in volume or it can be higher in volume. So a gene could be partially turned on depending on what it needs to do at that time. For example, a good example again is puberty. A different set of events happen during puberty, and so all these genes are dormant till 14 or 15 years, and then they come into play and they're switched on. But it's not a binary situation. It can be the volume. I would think of it as a volume button. Mm-hmm. I do have the advantage of temporal heterogeneity. So even though they live close to each other, it's not the same time period because we have births that took place between 2017 to 2020. So the 9-month period are different 9-month periods. That being said, your question is very valid. I have very low levels of air pollution exposure here, of PM2.5 exposure here, because the year it generally tends to be pretty clean air here around here. And that is one of the things that I'm seeing in my analysis is most of them have low exposure, so I'm not seeing very like great difference between high and low levels of exposure. Yeah. Mm-hmm. Right. Mm-hmm. Mm-hmm. Yeah, no, yeah. No, that's a really good point. I am including a sort of like traffic variable close to highway, far from highway. I've coded it to be a binary variable right now, but maybe there's also a way to think about close, closer, further, far, very far, to get in a little more granularity. And I'll talk to you about this source, these maps that we're talking about later. Thank you. Sorry. Mhm. Well, purple air is I just showed it to you because it's really interesting. But a lot of these countries have their own monitoring stations and their own monitoring networks. But it remains a problem. The US has really good monitoring network. Australia has good monitoring networks, but some other countries in the world do not. But like I said, the satellite data, which is why that is so interesting, it is across the world, you know, and it can be a good proxy to use while we establish the monitor network. Yeah. No, I mean, go ahead. Yeah. Yeah, yeah, I don't know. Fresco. She's wearing a ball gown, then she gets royal jelly. No, I don't know. All right. So that was purple air. Right. So I touched upon this a little bit. So I do want to look at temporal analysis because um, at the time of fertilization, all the epigenetic markers from the parents are erased, and during the first week of life, they're again tagged back on. So it could be that the first week is more vulnerable than some other week during the pregnancy, or the first trimester is a more vulnerable period than something else. So I want to look at weekly, monthly, and trimester, just parse that out as well. I do have, because I just got this data from publicly available sources, I do have preconception data 3 months before conception that I can also look, although I don't expect to see a lot, but I have that data. That was my presentation, but I'm happy to take questions. Alma. Thank you. So we did not ask any questions related to air pollution in particular because this study was not designed for that sake. I just decided because this data is very difficult to generate, I decided that I should, try to look at this in this particular birth cohort. So we do have smoking data. We do have asthma data. We have all like cardiovascular diseases, diabetes, we have that data. We also have alcohol consumption data. So I'm just trying to include these variables, which on a theoretical basis, if they make sense and they might affect my outcome, I'm trying to use those variables. So for example, I'm using birth weight as one of my outcomes. And of course, I have to take into account gestational age for that. So I'm trying to, yes, that's a good question. I don't have historical data about where they were born, for example, and whether they were born in polluted areas. No. Robin?
That's all right. I have a sort of similar question about just sort of the treatment of confounding factors here, because it seems like something like air pollution would have so many other things that like come with that person's life and exposure to other things and diet and all these things. And then even I was thinking it was so interesting the example you gave about the Dutch famine and the effect on grandchildren and stuff. And so I'm curious, like even broader than your work specifically, but like methodologically in your field, like what are the ways of dealing with all of those confounding variables?
So, if, for example, I'm looking at air pollution, so I'm definitely going to take into account smoking. I'm also going to take into account traffic, how close they are to traffic or not. And then if the outcome is birth outcome, I'm going to take into account, the gestational age or the mother's BMI, because we know that the baby's gender, all these things. It's basically that you just try to draw for as many confounding things as you can. Yeah, so because I have medical abstraction data, I have basically their medical history, so I have already explored these things, but they don't seem to matter. Like diabetes does not seem to have an effect on the outcome. Hypertension does not seem to. I've been like playing around with all these variables and seeing if they are important to this model.
Got a couple people not thinking.
So I was saying that one of the very big problems is getting an estimation of exposure, and you might have this multivariable model which ends up with some kind of exposure. But the kind of exposure you have is multifactorial. It's the air, it's the smoking, it's the birth, it's all those things. So you've got a really tough problem there. And I'm wondering, do you have any results yet? And if you do, well, what results do you have so far? I'll stop there.
So I have seen some effect on preterm birth. So the higher the levels of exposure, the higher the chances of a preterm birth. So that is one outcome I have seen. Now coming back to DNA methylation. When I say I have DNA methylation data, what I have is this, whether a site was methylated or not, for 850,000 sites on the DNA. So one way to look at this is look at each one of these sites and see if it was associated with air pollution. Basically do 850,000 linear regressions and see if anything comes out. I have done that step. I did not see any of these sites as associated with air pollution. Yeah, so my data for each site, I get a value between 0 and 1. Usually every site is either it's methylated or it's not methylated, right? And a gene has several of these sites, right? Yeah.
I don't know.
Yeah.
I'm ignorant of the thing, so I don't know how you exposure air or environment and go from that.
It's a statistical model. So I get this I get this data from, it's called Illumina Epigenetics. It's this whole platform that gives us this DNA methylation data. And then I have my DNA methylation data, I have my exposure data, and I just run linear regression. Yeah. Yeah. A lot of this is already figured out.
About that. What genes did you choose and how many are you looking at?
So I have not started looking at genes yet. Oh, so if I can go back to one slide here. Right. So let's say from here to here is one gene, right? But the data I get is for, like, I get a value for this rung of the ladder and this rung of the ladder. So I start by looking at just one site. These are all called sites. I start by looking at one site, and I have 850,000 of these sites. Now the next step is differentially methylated regions. Instead of looking at one site, you look at a region. And these regions are defined, I forget right now, but they're defined by the number of base pairs they have. They're defined by the number of these sites that they have. And I have looked at that and I did not find any differentially methylated regions. But then I started to look at genes, just comparing the low exposure group with the high exposure group. And I did find some genes that were very broad buckets, like something like something related to placenta. It's a very broad bucket of genes, but I did find some broad buckets of genes that were different between the low exposure group and the high exposure group of air pollution.
Thanks, Reshma. Oh,
Oh, I'm sorry. I just saying I wanted to applaud your courage in trying to attack a problem like that has so many, so much fuzziness to it in various areas. But
Yeah,
good on you.
I just want to say something about that. I might not see any great positive groundbreaking results, but it's important to still do this kind of study because you can say something about the exposure, like is satellite data good to use for a study like this? Or you can say something, North Carolina is great, you don't have to worry about PM 2.5. You can always say something even if you don't find like a positive outcome and you don't want it to have an effect.
yeah, actually, Reshma, that was kind of my question. And epigenetics, first of all, epigenetics is a quite new field. Is that correct? Is that a fair? Not really. Okay, but it seems like there's a lot of unanswered questions, especially this link between, like for example, low birth rate and non-communicable diseases, which is what you're trying to study. What that link is, if I'm understanding correctly, and you explained that there are many different hypotheses out there, right? So you're exploring the methylated, but depending on what you find, as you were kind of just articulated, and let's say you don't find what you thought you were going to find, that will help the field to rule that option out then, and thereby still progressing forward in ultimately finding out what that link is. And that's a really amazing thing that you're doing with your research.
Thank you. I still haven't finished my analysis. I haven't included psychological stress data and financial stress data. Maybe when you include those, we might see something else. There is another statistical model called distributed lag model, so I'm using monthly PM 2.5 data. So month one, these are not independent values. Month one is actually related to month two to month three because the seasonality of exposures. So this distributed lag model takes care of the fact that these are not independent exposure values but are related to each other. So there are a few more things that I could try still to see.
I'm formulating my question as I stand up. it's really interesting to compare this talk today to the talk last week, which was, it's very much about visual studies. How hard is it to talk to a lay audience about scientific hypotheses, processes? Is the expectation of ignorance among the audience like something that you just have to go in with?
No, I don't find it hard at all because I was where you are a mere 2 or 3 years ago. So all this information is new for me too, and I have learned it along the way. And I actually like talking to people about my research and explaining about my research. The only thing sometimes that I struggle with is as you get deeper and deeper into your own work, you don't realize what questions the audiences might have. And so I like to get the audience questions and it's like, okay, I should have explained that. Of course I should have explained that, but it does not occur to you when you're explaining that that is a gap that could exist and which is always interesting to me. Thank you. Thanks. Thank you for your time.